Hey everyone, this is Digestive System video number 8, the next to last video. So this video is going to cover the liver, anatomy and physiology of the liver. And so on the outline here, we've covered all of these. We've skipped over large intestine, covered these, and this is the liver video. And then we'll come back in one last video having to deal with the large intestine. All right, some fun facts about the liver. First of all, it is the largest gland in the body. It's not as big as the skin, but and if you consider only the glands inside the body, the liver is the largest. The liver is sectored off into 4 lobes, and these lobes are defined by a connective tissue barrier, I guess you could say, between them. So from the anterior view, you can see the first 2, right and left. And then a posterior view will allow us to see the other 2, caudate and quadrate. From an anterior view, you can see this falciform ligament, and that is basically the divider, connective tissue divider between the 2. So here is an anterior view of the liver. First of all, liver, upper right abdominal quadrant up against the bottom of the diaphragm, and it's really well protected by the rib cage, mostly covered by the rib cage. Anterior view, you can see the right lobe. This is the largest lobe of the liver. Left lobe, and the typical shape is sort of a triangular shape to it. The gallbladder, you can see that poking down on behind the right lobe. And this is connected by ducts to the right lobe. Some associated structures to the liver. So we talked about the mesentery that attaches the liver to the stomach. That's the lesser omentum. And there's a structure or a location on the posterior surface called the porta hepatis. This is where the blood vessels enter into the liver. Main place that they enter. Okay, the gallbladder, as we mentioned before, is on the posterior surface of the right lobe. I think this might have been miscorrectly stated in a previous video. I think I said it was on the ventral surface. It's not. Dorsal surface, posterior surface of the right lobe. Okay, this is a posterior view of the liver, and here is the right lobe, the largest. Here is the left lobe. And then from the posterior view, you can see the 2 other lobes, relatively small, caudate, superior, and quadrate, inferior. Here is the gallbladder. The screen here is covering that a little bit. Oops, sorry, let me go back. Gallbladder. And these are some of the ducts connecting the gallbladder to the liver. Another structure here. This is the porta hepatis. This is the entryway for gallbladder ducts and also blood vessels into the liver. All right, blood circulation to and from the liver and within the liver is kind of complicated. Basically, in terms of macroscopic vessels connected to the liver, there's a hepatic artery, and this branches off of the descending aorta, and delivers oxygenated blood into the liver. This is going to divide extensively inside the liver, and it forms hepatic arterioles, and that's delivering oxygen-rich blood to the hepatocytes. Blood from the liver is drained into hepatic veins, and these are going to connect into the inferior vena cava. And the hepatic veins are connected to what are called central veins in a microscopic structure called the liver lobule, which I'll mention in a bit. There's one other main vessel connecting into the liver, and this is a vessel that's delivering blood directly from intestinal organs. Small intestine, large intestine, also the stomach and the spleen. So vessels draining blood from those structures merge into this hepatic portal vein, And all of that blood from those structures goes straight to the liver. Okay? And then inside the liver, that's going to divide into hepatic portal venules, which is going to deliver this blood to the hepatocytes in the liver. That's kind of a unique delivery system, so let's take a look at a short video describing that. Hello, so this is a short video to describe blood circulation to and from the liver, and also within the liver. So it's a little bit complicated, unusual type of circulation, and I want to describe this both at the macroscopic level and at the microscopic level. So let's start here with the macroscopic level. Here is the liver. Blood circulation to and from the liver is typical in terms of two of the main vessels involved. There's a hepatic artery which branches off of the descending aorta, and that's going to bring in oxygen-rich blood into the liver. All right, that's typical for any kind of vessel. And also the liver is drained by what's called a hepatic vein. There's a set of hepatic veins. I'm just indicating one on this drawing. And that's going to drain oxygen-poor blood from the liver, right? So the cells in the liver, the hepatocytes, get oxygen from the hepatic artery, and they circulate away oxygen-poor blood through the hepatic vein. The hepatic vein is going to connect to the inferior vena cava, bring blood back to the heart. So far, so good. That's typical. Here's where it's atypical. There's another vessel bringing blood into the liver. This vessel forms by merging of vessels that drain the intestines. Small intestine, large intestine, also part of the spleen and the stomach. So there's a huge network of blood vessels from all of those organs that are going to merge into one main vessel called the hepatic portal vein. This is the third of the three major type of vessels involved. This hepatic portal vein carries oxygen-poor but nutrient-rich blood. It's nutrient-rich because it's coming from the intestines that have absorbed nutrients into the bloodstream. Okay? So that's the macroscopic level. Now let's shift to the microscopic level. At the microscopic level, the liver is filled with structures called liver lobules, basically hexagonal structures that are filled with hepatocytes. So all these purple squares here are meant to represent the hepatocytes. And at each of the 6 corners of this hexagonal structure, there are 3 vessels. That's called a portal triad. The portal triad includes one bile duct, I'm representing in green here. One of the functions of the hepatocytes is to make bile, and that bile is going to enter into this bile duct, and it's going to flow away, and eventually it's going to leave the liver. And it can go to the gallbladder or to the duodenum. Okay, the other 2, color-coded red and blue. The blue is a hepatic portal venule. This is going to be a branch of this hepatic portal vein. It's going to enter the liver, and then it's going to branch into this hepatic portal venule. The hepatic portal venule is bringing oxygen-poor, nutrient-rich blood to the hepatocytes. They're going to process that blood. Basically, they're going to filter out the nutrients, and they may store some of them. Like, for example, glucose can be put into glycogen, or they may use some of them. For example, amino acids can be used to make a number of proteins that hepatocytes are involved in making. Okay? The hepatic arteriole is a branch of the hepatic artery. So this is bringing oxygen-rich blood into the hepatocytes, supplying oxygen mostly. Okay? These 2 vessels, hepatic portal venule and hepatic arteriole, are going to allow their blood to filter through these hepatocytes in the very leaky liver capillaries, the sinusoid capillaries. So blood from both of these is going to go into the sinusoids, and it's going to filter through all of these hepatocytes. And then eventually this blood is going to collect in a central vein that's running through the center of every the liver lobule, and that's going to drain the liver lobule, and this blood is going to go to the hepatic veins and then circulate away in the inferior vena cava. Okay, so again, the circulation, two vessels bringing blood in, one oxygen-rich hepatic artery, one nutrient-rich hepatic portal vein. Those are going to branch into, at every of all 6 of the corners of the liver lobule, a hepatic portal venial, hepatic arteriole, and that blood is going to filter through the hepatocytes in the liver lobules, very leaky capillaries, allowing the hepatocytes to process both types of blood. After processing, the blood is going to collect in the central vein and then circulate away from the liver. So this is kind of the intricate circulation present in the liver. All right, so that's the circulation. And I have a few slides just to review that and, with better illustrations than what I could draw, basically. So this is the portal circulation. This is indicating all of the intestines, well, actually not all of them, parts of the intestines, stomach, spleen. They are sending vessels that contain blood from each of those structures, and they're all going to merge into this one structure, hepatic portal vein. That's going to enter into the liver, and it's going to branch extensively in the liver, and this is going to deliver this nutrient-rich blood into the liver and be processed by the hepatocytes. Okay, that's the hepatic portal circulation. Now we're inside the liver and looking at the microscopic anatomy of the liver. And if you section through the liver, put that on a slide, you will see something like this. This is a liver lobule in cross-section. It's defined hexagonal by this hexagonal border, which is a connective tissue border. It's a septum, and you can see the 6 corners of each lobule. This is a cross-section, but within the liver it would be a hexagonal column, basically. Okay? In the center of the lobule is the central vein. In this slide it just looks like a space, but that would be where the vein is located. And then surrounding the central vein, these are hepatocytes. Okay? And this is just showing that it's a repeating structure inside the liver. So this is the main structural organization that you find in the liver, these hexagonal columns called the liver lobules. All right, a little bit more about that now. So this is sort of a 3D representation of a lobule, and you can see it's a column here. And at the corner, each of the 6 corners, you find the portal triad, which is made up of one bile duct, indicated in green, one hepatic arteriole carrying oxygen-rich blood, and one hepatic portal veniule carrying nutrient-rich blood. These are going to connect to the liver sinusoid capillaries, and blood from these 2 vessels are going to filter through the hepatocytes and then eventually to make it into the central vein. And then they're going to circulate away from the liver lobule. For the bile ducts, hepatocytes make bile, and the bile ducts, bile that's made flows toward these ducts. So blood is going to flow inward toward the central vein. Bile is going to flow outward toward the bile ducts at the 6 corners of each lobule. Okay, and this is just showing the portal triad again, each of the 3 vessels, and the connection to the sinusoids for blood vessels, and the connection to the bile duct for bile production and transport. Right, and then this is just showing a few liver lobules grouped together. The neighbors share walls. There's one of the structures shown on this, and that's a bile canaliculi, or singular would be canaliculus. These are fine channels, and these are extensive throughout the lobule, and you find a lot of these between hepatocytes. So the bile that gets produced enters into these small vessels, canaliculi, and they're transported to the main bile duct at the 6 corners of the lobule. Right, and then this is just kind of a magnified view showing same structure, but blood entering either of these two. So hepatic arteriole is going to enter into the liver sinusoids, and hepatic portal venule, also sinusoids, and is going to be filtering through the hepatocytes. Oops, sorry about that. So bile, on the other hand, is flowing in the opposite direction through all of these canaliculi into the bile duct. Okay. What are the main functions of the hepatocytes? So let's summarize those. Main functions. Hepatocytes do a lot of things. One is bile production. So bile is produced in the liver, stored in the gallbladder, and used during digestion, especially of lipids. Another is processing of bloodborne nutrients. And I gave you a few examples. When you eat and absorb glycogen, eat and absorb glucose, it can be stored in the glycogen in hepatocytes. Hepatocytes can also use absorbed amino acids to make a long list of plasma proteins that they make. So that includes albumins, clotting factors, etc. Hepatocytes store fat-soluble vitamins, vitamin B and K. And finally, hepatocytes detoxify ingested toxins. So that's important because stuff that's absorbed through your digestive tract is transported directly. First stop is straight to the liver. And if there are any toxins, they can be detoxified. And if there are any nutrients, they can be processed. Right, so I'm going to end the video here, and next time the last video is going to cover the large intestine. See you later.